Hello, my name is Ava Homan, and I am presenting on the impact of different diet patterns on the gut microbiome. So, starting with the bluff, as more people adopt plant based diets, it's important to understand how these choices influence our health at a microbial level. And specifically, the gut microbiome has been shown to have influence on digestion, immunity, and overall health. So, the research questions that the researchers were trying to address were how do vegan, vegetarian, and omnivore diets impact the structure and function of the gut microbiome? And what are the health implications of any microbial differences between what they find? And also, they were curious to learn if they can use the microbiome data to predict someone's diet pattern with machine learning. And by answering these questions, they could help improve dietary recommendations and public health policies. Um, and so this study identified distinct microbial signatures associated with each diet, and they demonstrated that these dietary habits strongly influence the gut microbial diversity and composition. So, to give a more in depth answer as to why we should care about this topic, the gut microbiome is crucial for many aspects of health, from digestion to immunity. And what we eat directly affects which microbes thrive in our gut. And this has major implications for conditions like diabetes, heart disease, and obesity. And also, with plant based diets becoming more popular in recent years, it's crucial to examine how different dietary patterns shape the microbiome. So, the field of microbiome research has grown rapidly throughout the past 20, 30 years with advances in sequencing technology. And so past studies have been limited in size, but this research article was able to analyze over 21,000 people. So it provides a broader and more accurate picture of diet related microbiome differences. And these findings are helping to shape personalized nutrition and public health policies. This study has made several important contributions. It identified clear microbial differences between diet groups and showed that food microbes can transfer into the gut, and emphasized the importance of plant diversity in shaping a healthy microbiome. These findings also provide valuable insights for both researchers and dietitians. And this research is urgent because diet related diseases are on the rise worldwide, like cardiovascular health and diabetes, obesity. And so, understanding how diet affects the microbiome could lead to personalized nutrition plans that may improve health outcomes. And this work also has broader implications for public health, sustainability, and even agriculture. So, for the methods of this study, they analyzed microbes from 5 different cohorts, totaling over 21,000 participants, and they used 3 large-scale Zoe Predict cohorts from the UK and the US, and then 2 smaller data sets from Italy were also used. And omnivores made up the largest group, followed by vegetarians and then vegans. So they provided dietary information as to, what their diet looks like, as well as stool samples. And from these samples, DNA was collected using shotgun metagenomic sequencing techniques, and they used the Illumina NovaSeq 600 platform in order to analyze the data. And so this profiling, they were able to determine the species level abundance, and they fitted linear models for analysis of these differences between the diet pairs, and then conducted meta-analysis to combine these results, and Permanova and beta diversity metrics were used to analyze the microbial composition across diets. And they also did machine learning using random forests to predict diet patterns based on microbiome composition. So for this first figure, figure 1A is just showing the sample sizes in the 5 cohorts that are listed on the x-axis, and the sample size is in each cohort is color-coded by the diet pattern that was recorded by the individuals in each cohort. And in figure B, this is denoting the alpha diversity of the samples. So it's the observed richness plotted against each of the 5 cohorts, and it showed that it was highest in omnivores and significantly lower in both vegetarians and vegans. And there was no significant difference in microbial richness between vegetarians and vegans. And so this data suggests that excluding animal-based products reduces microbial diversity. For 1C, this is looking at the Healthy Plant-Based Diet Index, and this is scoring dietary patterns based on their intake of healthy plant-based foods versus unhealthy plant or animal-based foods. So higher scores went to healthier plant foods, and lower scores were to less healthy plant and animal foods. So the result was that vegans had a higher HPDI, followed by vegetarians, and in last place, this was omnivores. And I want to mention that this is just correlations, and it's not necessarily indicating that people who eat omnivore-based diets are less healthy, but yeah, because there's more microbial species that show up in from eating vegetables, which vegans and vegetarians assumedly eat more than omnivores, which is why these values are differing like that. So lastly, figures 1D through H are the plots of the beta diversity using unweighted unifrac distances, and these plots, principal coordinate analysis (PCOA) plots, they show clear clustering of gut microbiomes by diet pattern, and it shows that all 3 diet patterns exhibited distinct gut microbial compositions across all cohorts, and it confirms that dietary habits significantly shape gut microbiota. Okay, so this figure and the next 2 figures are kind of related. It's a comparative analysis of the gut microbial species level genome bins, or SGBs, between omnivores and vegetarians on this slide, and then the next slide will be omnivores versus vegans, and then the last slide is vegetarians versus vegans. So for this slide, the key findings of this are we are seeing the microbial signatures of omnivores versus vegetarians, and so we can see that omnivores had a significantly higher prevalence of specific bacterial species compared to vegetarians, and the strongest microbial associations are found in omnivores, and this suggests that animal-based foods are contributing unique microbial taxa. And so a total of 488 SGBs were differentially abundant in omnivores compared to vegetarians. And along with this, meta-analysis was conducted to assess the correlation between gut microbial abundance and consumption of the 5 major food groups, including red meat, white meat, dairy, fruits, and vegetables, which is under the C part of the figure. And this showed that omnivores showed strong positive correlations with microbes linked to meat and dairy consumption, obviously, because they eat meat and dairy. And then vegetarians are showing a higher level of fiber-associated bacteria involved. And this is involved in plant polysaccharide fermentation. Now, this slide continues with the previous slide, but now it's comparing the omnivores versus vegan diet. And from this data, 626 SGBs were differentially abundant in omnivores compared to vegans, showing the increased difference of microbes that were detected, or species that were detected. And so this also shows that meat and dairy consumption strongly correlates to specific microbes showing up in the gut microbiota. Yeah. And this final graph shows the microbial differences between vegetarians and vegans. So for this, only 11 SGBs were differentially abundant in vegans compared to vegetarians. So this highlights the similarity between these two diet groups. And so we can see that dairy-associated bacteria such as Streptococcus thermophilus and Lactococcus lactis were significantly more prevalent in vegetarians due to dairy consumption. And vegans exhibited a higher abundance of plant-associated microbes, including those found in fermented foods. And so for the predictive modeling, the machine learning models using the dietary information were able to predict microbial presence with moderate accuracy, with the area under the curve AUC values ranging from 0.62 to 0.72. So this indicates that while diet explains some microbial variation, there are other factors that likely contribute. And so the primary differentiator between vegans and vegetarians is dairy consumption, with a measurable impact on gut microbial composition. And even though both diet groups have high fiber intake, removing dairy from the diet appears to shift the microbiome further towards a plant-exclusive profile. And this highlights the importance of specific food groups rather than broad dietary categories in shaping the gut microbiota. And so overall, out of all 3 of the data sets that were given comparing between the diets, omnivores exhibited the most distinct microbiome signature, with microbial species strongly associated with meat and dairy intake. And vegetarians and vegans shared many more microbiome similarities, but dairy consumption played a key role in distinguishing their microbial profiles. And so this reinforces the link between diet and microbiome composition, implicating possible dietary recommendations or gut health and personalized nutrition strategies in health. So this next figure shows how food-derived microbes contribute to the gut microbiome composition across different diet patterns. Um, and so panel A is showing the cumulative relative abundance, the log of that, of food-associated microbes in the gut. Um, And B is showing the number of food species genome bins that are detected in each individual's microbiome and categorized by diet pattern. And C shows the prevalence of the 20 most common food associated microbes across the 3 diet groups. And finally, panel D is showing the distribution of the 20 most common food microbes across different food categories, which is including meat, dairy, fruits, and vegetables. And so the findings of this, or what this implies, is that there's greater food microbe transmission in omnivores. So omnivores are having a significantly higher cumulative abundance of food-associated microbes compared to vegetarians and vegans. And the number of detected food-derived SUBs was highest in omnivores, followed by vegetarians and then vegans. And so the asterisks for A and B are indicating the statistical significance, with 2 asterisks denoting a p level less than or equal to 0.01, and 3 denoting a p-value of less than or equal to 0.001, which is showing highly significant differences between these groups. And so the diet-specific microbial signatures, dairy-associated microbes, including Lactobacillus acidophilus, Streptococcus thermophilus, and Lactococcus lactis, were predominantly found in omnivores and vegetarians, but almost absent in vegans, which makes sense because vegans are not consuming animal products such as dairy. And plant-associated microbes such as Enterobacter hormacae, Citrobacter frundi, and Raulatella ornithinolytica, and Klebsiella pneumoniae were most prevalent in vegans, which is supporting the idea of food-to-gut microbial transfer from plant-based foods. So some of the microbes that were found in the vegan gut microbiomes were also common in soil and agricultural environments, which suggests that plant-based diets might introduce distinct environmental microbes into the gut. And so this supports the idea that microbes from consumed foods can become part of the gut microbiome, with omnivores experiencing the most direct microbial transmission. And it also is noting the impact of the diet on microbiota, with plant-based diets introducing different microbial profiles compared to meat and dairy-heavy diets, which might have implications on gut health, which include dairy-associated microbes are often linked to probiotic benefits, especially fermented dairy, such as yogurt and cheese, but there's also fermented products such as kimchi for vegans that are also linked to these probiotic benefits as well. And soil and plant-derived microbes could play an unexplored role in gut immune function. And so overall, this highlights the intricate relationship between diet and microbial diversity, and provides further evidence of the direct impact of food consumption on gut microbiota. So this last figure is demonstrating the ability of machine learning models to predict diet patterns based on gut microbiota. Um, so a random forest classifier was used to distinguish omnivores, vegetarians, and vegans based on microbiome data, and it achieved an average AUC of 0.85, which indicates strong predictive power. And it was best, its best prediction performance was between omnivores versus vegans, with an AUC of 0.9. And then lower accuracy was found between vegetarians and vegans because of the similar microbiomes with an AUC of 0.84. And then omnivores versus vegetarians with an AUC of 0.82 was the lowest reliability. And so this implies that the microbiome composition can reliably indicate diet patterns, and machine learning models could be used to help identify microbiome-based dietary biomarkers. And some classification errors may result from recent diet changes not yet reflected in gut microbiome biota in this. And so overall, this is showing that diet is a major driver of gut microbiome diversity. And machine learning models can effectively classify dietary habits based on microbiota composition, which reinforces the strong link between diet and gut health. And this highlights the potential for microbiome-based dietary assessments in clinical and nutritional research. So overall, the key results are that this study showed that vegans and vegetarians had lower gut microbial diversity compared to omnivores, and each diet group had distinct microbial signatures, with vegans having more fiber digesting bacteria showing, while omnivores had more protein fermenting microbes present. Additionally, there was strong evidence that microbes from food contribute to gut composition. So to summarize, diet has a major impact on the gut microbiome, and this in turn affects health. The research identified clear microbial differences among diet groups, with plant-based diets generally associated with a healthier gut microbiome. These findings have important implications for dietary guidelines, public health strategies, and the potential for personalized nutrition plans tailored to microbiome composition. Future research should focus on strain level analysis and diet microbe interactions over time. Okay, so the discussion questions that I would like to talk about that I came up with are: how do gut microbial differences between omnivores, vegetarians, and vegans affect health outcomes? What are the potential challenges in translating microbiome research into personalized dietary recommendations? Could dietary interventions targeting gut microbiota be used as a therapeutic approach for metabolic diseases? How do cultural and geographical dietary differences impact microbiome composition in global populations? And finally, what are the limitations of current microbiome studies, and how can future research address them? Thank you for listening, and here are my references, and I am looking forward to discussing this during the next journal club.